Okay guys, so you can see there are certain kingdoms, okay, that has been classified as per our lineage. In Plantae and Animalia, they placed group what? Algae, fungi, bacteria, bryophyta, pteridophyta, spermatophyta. And if you talk about Animalia, we have coelentrates, okay, then it's Polydera, okay, I think, yeah, it should be F, by mistake they mentioned D, Porifera. Platyhelminthes mollusca, okay, Nemathelminthes annelida and echinoderms. Now problem is that, what is the problem? Like first thing I said, that if we talk about fungi, okay, so fungi, they have cell wall, okay, but they are saprophytes. So my problem is very simple, that how we can place them in plants. Logical. Now one more thing, if you come here, yeah, poriferous is fine. If you talk about amoeba will also come in Animalia, okay, they are placed in this thing. Now here if you talk about bacteria, okay, fungi, algae. Bacteria is our what? Bacteria is our prokaryotic organism, okay, you very well know this. And remaining, if you see all the groups, algae, fungi, bryophytes, pseudophytes, they are what? They are eukaryotic. So how we can place prokaryotic and eukaryotic organisms together? The problem is that if we are making groups, we have to make groups properly. We cannot just mix them in just one character. One more thing, amoeba is unicellular organism. Amoeba is unicellular organism, and you can take any other example. They all are what? They all are multicellular. Okay, same problem you can see in case of plants also. Few of them, they were unicellular, they were placed with multicellular. So there were certain problems, and when we have a problem, we used to go for the new system. So now again I'll repeat guys. First point that we have to remember is that why we need classification. Second point, we have to remember about the classifications of Aristotle. He gave for the animals and plants. Now we came to third point, and that third point is who gave the two kingdom classification. That was given by Carolus Linnaeus. Name of scientist is important. This all things are not important. Whatever mentioned or written by pen, that is important. And same thing is here also. Eukaryotes and prokaryotes, they were not classified. Unicellular and multicellular, they were in the same group. Photosynthetic, non-photosynthetic, they were also in the same group. Very important. Besides morphology, as a need was also filed for including what? Characters like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, and evolutionary relationships. So initially, scientists were talking majorly about morphology. So it was required that we should include what? We should also include other characters like cell organization. So if we talk about next is our this thing, Ernst Haeckel. Ernst Haeckel, he gave three kingdom classification, right? So what he did, Ernst Haeckel, he introduced our kingdom Protista. So with the Protista, we can say kind of cell structure, or we can say cell organization, because Protista, it includes all unicellular organisms. Let's come here. Yeah. So three kingdom classification was given by Ernst Haeckel, and he introduced kingdom what? Protista. And Protista include all the organisms which are unicellular, okay, and one more important thing, which are eukaryotes. Okay, so now the point is important guys. As of now, if you don't want to mention eukaryotes, fine, but it's okay. Unicellular eukaryotes. Now what is benefit for us? Now we have Protista, it includes all unicellular organisms, okay, and it includes all the eukaryotes. So the problem of multicellular and unicellular being together, it has been separated, right? But there was one more problem, and that problem is what? Photosynthetic and non-photosynthetic together, and our prokaryotes and eukaryotes together. So after this Ernst Haeckel, we had next scientist, that is what? Copeland. And Copeland introduced a new kingdom, that is our what? Monera. and Monera included all the organisms which are prokaryotes. So this is our solution of next problem, that is now prokaryotes has been separated from all the groups and they have been placed in the single group, that is called Monera, or we can say bacteria, right? So first problem was solved by Unstackel and third problem was solved, sorry, second problem was solved by our Copeland, and third problem it was solved by R.H. Whitaker. Okay, what he did, he gave the next sites. So the problem of being fungi and plants together, that is also resolved, right? So now what the thing is that I am not saying that you have to remember all the points. Whatever you have to do is we have to remember the name of scientist, which type of classification he gave, 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, which kingdom was introduced by that particular scientist, and what were the drawbacks. For example, in 2 kingdom there were drawbacks of unicellular and multicellular being together, prokaryotes and eukaryotes being together, photosynthetic and non-photosynthetic being together, right? But by the third kingdom classification we solved some problems. We separated what? Unicellular organisms. So one problem has been solved. Okay. Fourth kingdom classification was given by Copeland and he introduced the kingdom what? Our Monera. It will include all the unicellular what? Prokaryotes. So unicellular organisms separated, prokaryotes separated, eukaryotes separated, and the fifth kingdom was given by our scientist R.H. Whitaker. He introduced what? Kingdom fungi, and that separated what? Our photosynthetic organisms from non-photosynthetic organisms. Very simple thing, right? Very simple thing. It's not complicated. Just remember the key points. Now, in exam, a very popular question or very common question that examiner used to ask, that what were the methods, what are the basis on which R.H. Whitaker, okay, he gave the classification. And guys, if you ask me, generally, out of all the 3 systems, all the systems we discussed, 2, 3, 4, 5, so 2 and 5th is super important. Okay, if 3 and 4 name of scientist given in NCERT, fine. If not, you can ignore it. But 2 and 5 are super important. So what were the criteria on which kingdom classification, 5 kingdom classification was given? It was cell type, okay, prokaryotes or eukaryotes. Cell wall is not that much important, but it is mentioned here. Nuclear membrane, body organization, mode of nutrition, and one more thing was there, that is our phylogenetic relationships. Okay, so I'll open NCERT. I'll tell you what the keywords we have to remember. Just give me 1 minute, because we don't have to require all the things. Yeah, that's it. But this table is important. I'll come to this table again, but I want to just mention few things as per NCERT. Yes, so I'll highlight that point. You can see the main criteria used for classification by him includes what? Cell structure, okay, whether the organism is prokaryote or eukaryote, okay. Then body organization, unicellular or multicellular, mode of nutrition, okay. Then we have reproduction, and last is phylogenetic relationships, okay. So what is not included? If we come here, what is not included? It is not including what our, we can say, you see, if you talk about this thing, cell wall and nuclear membrane, collectively it will give the idea of cell organization only, so fine, no issues, but you remember this point: cell structure, okay, cell organization, cell structure, prokaryote or eukaryote, or body organization, unicellular or multicellular, mode of nutrition, reproduction, and phylogenetic relationships. So these were the criteria, okay. If in exam cell wall is given, it will not be correct. Okay, if in exam nuclear membrane will be given, it will not be properly correct. So we have to be very specific when we are saying cell structure. That means we are talking about prokaryotes and eukaryotes. Okay, and when we are talking about body organization, that means we are talking about unicellular or multicellular. Okay, see, everything's classify basic parts. First one, if you talk about Monera, Monera includes what? Prokaryotes. That means cell structure. Body organization means eukaryotes, unicellular. Monera also includes unicellular. Then mode of nutrition, fungi. Fungi have different mode of nutrition compared to other this thing. Then mode of reproduction, it can be different in case of plants and all. Okay. And last is mode of nutrition. Sorry, nutrition is done. Reproduction. So how they are reproducing and all. And last is phylogenetic relationships. What are the evolutionary relationships? Phylogenetic means evolutionary relationship between the organisms. Now let's come to this table again. It's not difficult, it's very easy, so will not take too much time here. And major character of our, all the kingdoms will be covered from here only. Cell type, Monera is prokaryotes, remaining all what? Eukaryotes. Cell wall, in case of Monera, it is made up of what? Non-cellulosic, they are mentioned what? Polysaccharides. Achha, to be specific, there is one more classification system that is called 6 kingdom classification. So in that, what we have done, we have classified our Monera into archaea bacteria and eubacteria. I'll come to that part also because they have certain domains. Okay, so archaea and eukarya and this thing. Okay, prokarya and eukarya. So I'll come to that part also. There are 3 domains, and sometimes, like I don't remember recently, I think past 6-7 years ago they have asked about those 3 domains. So I'll be covering that part also. So let's come here again. So in case of Monera, we have archaea bacteria and eubacteria, you very well know. In archaea bacteria, yes, we have cell wall made up of polysaccharides majorly. But if we talk about eubacteria, there we have a cell wall made up of what? Peptidoglycan. Right? Next, Protista, cell wall present in some. If we talk about initial groups, chrysophytes and dinoflagellates, they have a cell wall. Okay, but when we are going towards our euglenoids, so they have a protein covering, they have a pellicle. Then we come to slime molds and this thing, protozoans, they do not have cell wall, right? Fungi, cell wall of chitin, plantae, cellulose, and animals may absent. Nuclear membrane, obviously prokaryotes, they don't have nuclear membrane. Remaining all will be present because remaining all are eukaryotes. Body organization, cellular, because they are what? Or you can simply say unicellular or unicellular. Fungi is multicellular, they can form small tissues. In case of plants, we have organs also, but in case of animals, we have organ system and organisms too, right? Bacteria, they can be autotrophic, they can be heterotropic. Now chemoautotrophs, that means by chemicals, they can make what? Our energy and all. Heterotrophs, there are multiple things. If we talk about heterotrophs, so they can be saprophytic, that means they feed on dead and decaying organisms, just like our what? Fungi. They can be parasitic, feeding on other organisms. Protista, they also show multiple types, autotrophic and heterotrophic. In simple terms, if I say first initial 2 kingdoms, okay, they are what? Mix, auto and hetero. Fungi is completely hetero, plantae is completely auto, and animal is also completely hetero. Now, in case of heterotrophic mode of nutrition, guys, there are 3 conditions. What conditions are there? We have saprophytic, okay, we have parasitic, and we have holozoic, right? So if we talk about this thing, parasitic, that means feeding on some other organism, okay, some other organism that will be living. It will be what? Taking nutrition from from living host. Here from dead and decaying, and Holozoic when they engulf food. Okay, so the point is that what is very interesting in case of animalia, okay, animals, there is a nutrition given that is called saprophytic. So now you might be thinking that animals also show saprophytic mode of nutrition. Yes, a very basic and simple example you can take of earthworm. Earthworm, it shows what? Saprophytic mode of nutrition, right? So again I am saying guys, Monera and Protista, they show both type of nutrition, our autotrophic as well as heterotrophic. In autotrophs, we have further two types, chemoautotrophs, okay, or our normal autotrophs that can do photosynthesis. Chemo means chemical, auto means self-nutrition. So when organisms are generating energy from what chemicals, it will be chemoautotrophs. When they are depending upon other organisms, then it will be heterotrophs. Normal plants, they are simple autotrophs because they generate energy from sunlight. Fungi, saprophytic, and they can be parasitic also, right? If we talk about plantae, majorly what? Our autotrophs. So these are the 5 groups with some basic characters. I don't think it is complicated, and we all know since we are preparing for NEET exam that this table is very important. And after this table or below this table, this point is also important, that R.H. Whitaker, he used what? Cell structure, body organization, okay, and mode of nutrition, reproduction, and phylogenetic relationship for the what? For the our this thing, classification of 5 kingdoms, right? Yeah, so I was discussing, or I was planning to discuss this thing with you. Just a minute. Yeah. So guys, you can see here the 3-domain system has also been proposed that divides kingdom Monera into 2 domains. Like I said, archaea bacteria and eubacteria. Archaea means primitive bacteria, eubacteria means latest bacteria, leaving eukaryotic kingdoms into 3rd domain, and thereby there are 6 kingdom classification. What they are trying to say, let's come back to our notes. Just a minute. Yes. So this is our 6 kingdom classification. It has given what? Domain archaea. Archaea, we can say, you might be thinking that what is domain? A bigger group than kingdom, you can say, consider it as domain. So domain archaea, it includes kingdom what? Archaebacteria. We have domain bacteria, it includes kingdom what? Eubacteria. And we have domain eukarya, that includes kingdom Protista, fungi, plantae, and animalia. So domain, you can say, it is higher in hierarchy compared to the kingdom. Okay, so there were 3 domains, archaea and bacteria, and last is eukarya. Eukarya, very logical. All the eukaryotic kingdoms, they came in eukarya. Archaea, it also very logical. Archaebacteria, that means primitive bacteria, and eubacteria, we will have latest bacteria. Now you might be thinking that why we are classifying bacteria, very small organisms that we cannot even see. Why there is need of making 2 kingdoms, okay, for that small bacteria. Archaebacteria and eubacteria, guys, they show a lot of, we can say, dissimilarities. Their cell structure is different, okay, their habitat is different, their way of functioning is also different, right? So we initially thought that they both are prokaryotes, unicellular, so we can place them together, but their mode of nutrition, okay, it varies greatly compared to normal bacteria, okay. I think there is one more table that I wanted to show you, but fine, no issues, it's okay. Fine guys, so this is up to all the 5 kingdom classifications that we should know, okay, and it is important for us. Now the point is that we have to discuss about kingdom Monera, and kingdom Monera I will discuss from NCERT. I will not go beyond from NCERT because they are not giving anything. If required, I will add 2-3 points extra in NCERT, otherwise not more than that. In case of Kingdom Manera, what we have to discuss, again, in every kingdom we have to discuss basic things. Basic organization, right? Cell structure, like cell structure means shape and all, organization and mole, okay, mode of reproduction, and if there is any group and all that we have to discuss, and is there is the habitat and all. So basic things we have to discuss, right? Nothing more than that we will do. So if we come to NCERT, yeah, 5 kingdom classification is done. Yes guys, this paragraph is important. Okay, we'll go through this paragraph because there are some assertion based questions they used to give from this paragraph, so we'll not miss it. Okay, Kingdom Manera we can start later on also, but let's come to this paragraph. Okay, yes guys, so what they're trying to say, they are giving us just basic, drawbacks of 5 kingdom classification. See, in our NCERT we are reading about 5 kingdom classification, 6 kingdom classification, but when you will go in higher classes, okay, in your graduation, because obviously not in MBBS you are going to study the plant kingdom, you have to study the human physiology more, but when you go for graduation or something else, okay, so if you are staying in botany honors or something, so there you will study about this plant classification. So there you will see more branches and more groups, right? Now let us look at 5 kingdom classification to understand the issues and considerations that influence the classification systems. What are the benefits? First of all, earlier system of classification, they included bacteria and blue-green algae together, okay, fungi, mosses, ferns, gymnosperms, angiosperm, and the plants. Where in 2 kingdom classification, the characters that unified the whole kingdom was that all organisms included had their what? Cell walls in the cells. Logical. This placed together groups which widely differed in other characteristics. Okay. So obviously, like I said, in case of fungi, we do have cell wall, but it is heterotrophic. Bacteria, though some of them they are chemotrophs also or chemotrophs also. So their characters were widely different, but still they were what? Placed together. Next thing, it brought together prokaryotic bacteria and what? Prokaryotic bacteria and blue-green algae cyanobacteria with other groups which are eukaryotic. Okay, so prokaryotic bacteria, they were came were other eukaryotic groups, pteridophytes, bryophytes, angiosperms, gymnosperms, algae, they all are what? Eukaryotes. It also placed unicellular and multicellular organisms together. Chlamydomonas, Spirogyra, they were placed with higher plants. Okay, so these are just same characters which we discussed earlier only. The classification did not differentiate between what? Heterotrophic group fungi and autotrophic means green plants. So same 3 points. What? Prokaryotes, eukaryotes not differentiated, unicellular, multicellular not differentiated, and our complex organisms came with simple organisms, and our autotrophic, photosynthetic and non-photosynthetic, they were not differentiated. So that is why we did the classification, right? Next guys, what is the benefit of 5 kingdom classification and all? All the prokaryotes organisms, they were grouped together under kingdom what? Monera. And prokaryotic that we are talking about here, prokaryote is under Monera. And if we talk about eukaryotes, okay, they were placed under kingdom Protista. Kingdom Protista brought together Chlamydomonas and Chlorella, earlier placed in algae within plants, and both having cell wall with Paramecium and amoeba, which were earlier placed in the animals kingdom, lack cell wall. Now, this line is important and confusing also. Why confusing? Chlamydomonas and Chlorella, guys, you still study in algae, right? In plants. Yes, they are unicellular, but they are eukaryote. They can do photosynthesis. But here they are mentioning what? That kingdom Protistas brought together Chlamydomonas and Chlorella together. Like I said, there are multiple type of classification systems. Okay, there are some things which we have to study in higher classes. Usually in our 12th, 11th class, we study Chlamydomonas and Chlorella under what? Plantae. When you cover algae, there you will study it. Okay, there is no group that gives space for Chlorella and Chlamydomonas in case of our this thing Protista. But when you go in higher class, you will see that our Protista, okay, and this thing, sorry, that our Chlorella and Chlamydomonas are coming under Protista. When you go in higher classes, you will see one more thing, that cyanobacteria, in certain cases, it is included under algae because it is photosynthetic. It has same pigments compared to algae. But in some cases it is included under bacteria, right? So multiple things are there, but you can ignore this point. Just simply remember it, okay, as per this. Don't get confused in plant kingdom when you study Chlamydomonas and Chlorella there also. It has put together organisms which in earlier classifications were placed in different kingdoms, which is fine because they all are unicellular and they all are eukaryotic. This happened because sorry. This happened because the criteria of classification changed. This kind of changes will take place in future too, depending upon the improvement in our understanding and evolutionary relationships. Right? Over the time, attempt has been made to evolve classification systems which reflects not only morphological, physiological and reproductive similarities, but it is also phylogenetic. Phylogenetic means based on evolutionary relationships. So a simple point they are saying that over the period we are doing lot of improvement. We are working on bringing more changes. We are trying to make our classification more specific so that there should be no problems in placing organisms. Okay, so you should know that we are including morphological, physiological means functional, reproductive, okay, and phylogenetic means evolutionary relationships. This paragraph entirely same, but you should go through it, okay, and you should know Whitaker 5 criteria which were chosen for the classification. Now guys, let's come to Kingdom Monera. Kingdom Monera we are studying here, and about Kingdom Monera, or we can say bacteria, we used to study in case of our cell chapter also. Now I will give you a basic, this thing, run through that what all things are included here, so you will get a better idea. Yeah. So see, basic things. We have what are the shapes, then what are the features of bacteria, cell wall made up of glycolex, type of ribosomes, flagella and all, okay, and then we have to discuss about what our archaebacteria. That's it. Only this much is given in NCERT and we will not go beyond this, right? Now, so if I start with Kingdom Monera, basic things first. Yeah, basic characters you guys say omnipresent and all. Shape I'm not coming right now. Shape we know there are 4 types, coccus, perilium, bacillus and dot, other things are there. I'm coming to characteristics. So obviously they are prokaryotes, okay, and if they are prokaryotes, they will lack what? They will lack membrane bound organelles. They will lack membrane bound organelles. If they are prokaryotes, obviously they will have 70s ribosomes. Okay, if they are prokaryotes, obviously they will lack a nuclear membrane. Okay, and if you see here, this is our bacteria. They don't have nuclear membrane. DNA used to get accumulated in a particular region, and this region, it is called what? It is called nucleoid. You can also call it as genophore. Okay, and there is one more term for it, that is incipient nucleus. But my suggestion for you that you should remember nucleoid and genophore. Nuclear membrane I have not drawn. Okay. Now if we talk about prokaryotes, they have some membrane invagination. Now this membrane invagination, it can be in term of what? It can be in term of vesicles. Okay, it can be in term of some plate-like structures. These plate-like structures, they are called lamellae. It can be in terms of some tube-like structures. These are called tubules. So what I am trying to say, that if this is our bacterial cell, okay, I'll draw it again. If this is our bacterial cell, there is a possibility that membrane, it will form what? Invagination, either in form of tubes, either in form of lamellae, okay, or either in form of vesicles, right? If these vesicles, if these vesicles, they have what? Respiratory enzyme. So these vesicles, they will be called what? They will be called mesosomes. If these vesicles, they will have what? Photosynthetic pigments. It will be called what? It will be called chromatophore. Right? Now the point is, chromatophore, like I said, it will have photosynthetic pigments, so it will participate in photosynthesis. If we talk about mesosomes, so like we said, it has respiratory enzyme, so it will participate in what? Respiration. So kind of we can say it is prokaryotic mitochondria, but there are certain more functions of this our mesosomes, that is, it help in cell wall formation. Okay, it help in DNA replication. It helps in DNA replication, as well as it helps in DNA distribution, right? These invaginations, what they used to do, they increase surface area for metabolic reactions. Right? Now if we talk about extensions, okay, again here we have our bacteria. Okay, we have the largest extension, which is called flagella. Obviously it will participate in what? Movement. Okay, we have then villi, medium sized, okay, not this many, sorry, 3-4 I will draw. So this is our villi, help in majorly reproduction. Then obviously we have what? Small hair like fibers, and these are called what? Fimbriae, and it participates in what? Attachment, right? So these are the basic features which I have mentioned here, and I think this is enough. Obviously we have to make, we have to cover the shape part and nutrition part, so I'll come to that, but these are the basic features which we should know about our this bacteria and all. So now here also we can come, now we can go through all the points, okay, which are given in our slides too. So prokaryotic cell, genetic material is called what? Our nucleoid. Cell wall point I missed. Cell wall is made up of peptidoglycan, I mentioned in table, okay, it is also called what? Murin. And cell wall is made up of what? Or this peptidoglycan is made up of what? N-acetyl glucosamine or muramic acid. They will have mesosomes, okay, that will participate in what? Surface area, okay, help in cellular respiration, DNA replication or DNA distribution. Naked circular DNA, it is what? Plasmids are also present, and then they have flagella which participate in what? Movement and all, right? So comes this thing, cell envelope. If we talk about our bacterial cells, guys, we have cell membrane, okay, I'll change this thing. We have cell membrane, then we have cell wall, obviously made up of peptidoglycan. Above cell wall we have, here it is what? Cytoplasm part, cytoplasm or nucleus, you can say cytoplasm. And above cell wall we have what? Glycocalyx. Now glycocalyx is a third kind of, we can say, layer which is present in the bacterial cells. If it is very hard, it will be called, not mentioned here, fine. If it is very hard, it will be called what? Capsule. If it is smooth, okay, it will be called what? Slime. It will be called slime layer. This same thing will cover in our cell chapter also, so I'm not going into much detail here, but obviously you know this thing, Hana? Glycocalyx, hard, capsule, smooth layer will be slime layer, right? Now if we talk about shapes, so you can see spherical shape cocci, rod shaped bacillus, vibrio, spiral shaped, okay, spirillum, okay, they have mentioned image wrong, spiral, and it will come here. Vibrio will be comma shaped. So these are the four major shapes which we can see in case of our bacteria. You might have heard of two terms, that is strepto and staphylo. Strepto, wherever bacteria, they will be forming what? Chain. And wherever they will be forming or present in form of groups, they will be called what? Staphylo. For example, streptococcus. So bacteria, coccus means spherical. Spherical bacteria they form a chain. Streptobacillus, rod shaped bacteria will form a chain. Staphylobacillus, rod shaped bacteria will make a group. And staphylococcus, obviously rod shaped bacteria they will, staphylococcus, spherical shaped bacteria will form what? Groups. Right? I want to come to NCERT. Mode of nutrition I'll take from NCERT only. Okay. Yeah. So bacteria are sole member of kingdom Monera. They are the most abundant microorganisms and they occur almost everywhere. Hundreds of bacteria are present in handful of soil. Okay. They also live in extreme habitats such as hot spring, deserts, snow, deep oceans, everywhere. Bacteria are grouped together in the four categories on the basis of shape that we discussed. So I'm not discussing this again. Let's come to next part. Yeah. The bacteria are very simple. They have complex behavior compared to other organisms. And let's come to mode of nutrition here. some bacteria, they are autotrophic. That means they synthesize their own food from inorganic substrates, right? They can be photosynthetic or they can be chemosynthetic. Okay, photo means depending upon light for the nutrition. Chemo autotrophs means when they depend upon chemicals to generate energy by themselves. Inorganic substance means they are heterotrophs. Okay, chemoheterotrophs, when they depend upon other chemicals to generate energy, and the vast majority of them, they are the heterotrophs. That is, they depend upon other organisms on for food on dead end this thing, right? So they synthesize their own food from inorganic substance. They will take an organic substance, they will break it, they will get energy. Autotrophic. Now it can be of two types, photosynthetic or chemosynthetic. Photo mein they will use light, chemo mein they will use chemicals. If we talk about this thing, heterotrophs, they've missed one word, that is called chemoheterotrophs. They depend upon certain other chemicals also, and by utilizing it, by breaking it, they get energy. Okay, they oxidize those substances. For example, they oxidize iron, nitrogen and all, then they get energy from them, right? Next thing, guys, this is all about, we should know about the kingdom Monera. It's not given in NCERT also, all the points we discuss, because they have given same points in our cell chapter also, but basic points we should know about plasmids and other things we should know. And plasmid is not a new word, multiple times plasmid has been discussed in biotech and other things, so we should know about plasmids. Okay, same goes with flagella and pili and fimbriae. Flagella structure is there in the cell chapter, okay, properly given. Now if we talk about archaebacteria, in archaebacteria there are three groups. Okay, what are the three groups? We have halophiles. Halo means saline, salt. Phile means loving. We have methanogens, that is methane generating, and last we have is our what? Thermo means temperature, acid or acido means acid, philes means what? Again loving, PHIL means loving, right? So salt loving, methane generating and thermal and acid loving organisms. So these things, they come under our archaebacteria. Just a minute. Yeah, so these bacteria are special since they live in some of the most harsh habitats. They are extremely salty or extreme salty areas, halophiles, hot springs, thermoxylophiles, and marshy areas, they have methanogens. Now, what is the most important point in archaebacteria that we have to keep in mind is that they have different cell structure, they have different cell membrane structure compared to our normal bacteria or eubacteria. So archaebacteria differ from other bacteria in the cell structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in gut of bacteria, gut of ruminants also, such as cows and buffalo, and they are responsible for production of biogas or methane. Now guys, first point is, if we talk about cell wall, in bacteria it is made up of peptidoglycan, or we can say N-acetylglucosamine and muramic acid. In case of archaebacteria, cell wall is made up of polysaccharides, mentioned in table also. So cell wall is made up of what? Polysaccharides. Polysaccharides. Okay. Usually in case of bacteria or in any other organism, membrane have lipids like this, head and tail, they have lipids like this. But in case of, and this is what we are talking about here, is cell wall, and now we will be talking about cell membrane. In case of in case of archaebacteria, membrane, they have what? Branched chain of lipids. You can see how many hydrocarbon tails are there. So they have branched chains. Where? In the membrane. So structure of membrane is also different, and structure of, I should say, cell wall is also different. Both things are different, and that enables us our organisms to survive what? Extreme conditions compared to eubacteria. If we talk about eubacteria, very basic things that we discussed. Yeah, there are thousands of eubacteria or true bacteria characterized by the presence of what? Cell wall. They can have cell wall, they can be motile, and they can have flagellum. So, okay, cyanobacteria will come later. They can be photosynthetic autotrophs. They can be unicellular. They can live in the colonies. They can form the filaments. Okay, they are discussing about cyanobacteria only, not discussing about basic characters. It's okay, guys. So basic characters we have discussed. They're discussing about cyanobacteria. So cyanobacteria I'll tell you from here, from my slides. Okay, cyanobacteria, first of all, though nothing complicated. All the bacteria, they were what? Autotrophs and all, fine. Cyanobacteria, they were the first organisms which are capable of releasing what? Oxygen. That is a major point of cyanobacteria. Second major point is they were specialized cell that is called heterocyst. In that they can fix atmospheric nitrogen. That's it, nothing else. Okay, some of them they are free living, some of them they are symbiotic. Fine, but major thing is that cyanobacteria is the first bacteria that was capable of producing oxygen during photosynthesis. Earlier bacteria, like you mentioned there, photosynthetic autotrophs, they were able to do photosynthesis, but they were not releasing oxygen. So cyanobacteria were the first organism. Okay, they're the first organism to release what? O2, or they were the first prokaryotes to release what? O2. Cyanobacteria also referred to as blue-green algae because they have pigments. The blue pigment is called phycocyanin. The red pigment is called the green pigment is called chlorophyll. It's not getting erased. I'll cut it and I'll write it here. Yeah. Okay. And the other pigment is our chlorophyll. Okay. So they have phycocyanin and chlorophyll. Phycocyanin will give blue color, chlorophyll will give green color, and because of that they will have what? Blue-green color. They can be unicellular, single cell. They can be in form of colonies like this, or they can be filamentous like you can see here. They have a specialized cell that is called heterocyst that will participate in what? N2 fixation. Okay, photosynthesis does not take place in heterocyst, it takes place in other cells. These cells, they are called what? Vegetative cells. You can see entire filament is surrounded by a sheath of mucilage. Okay, so they are surrounded by mucilage sheath. Yeah, they are generally surrounded by gelatinous sheath. They are often form blooms in polluted water bodies. Polluted water bodies which have the pollution. Okay, because they can do photosynthesis, they can grow easily. In polluted water bodies, they will get nutrition also, like ions and all, minerals and all. So in polluted water bodies, I mentioned what? Blooms. Okay, that means they, or you can simply say they used to grow at very large speed. Okay, and they form algal blooms. Okay, some of these organisms can fix atmospheric nitrogen. We have example of nostoc and we have example of Anabaena. Chemosynthetic autotrophs bacteria, they oxidize various inorganic substances such as nitrates, nitrite or ammonia, and they use energy for production of ATP. Right? So chemosynthetic autotrophs, they were using inorganic substrates. So in this condition, O2 was not released, but here they are doing proper photosynthesis. They are using sunlight and all. Okay, so O2 is released there. They play a greater role in recycling nutrients like nitrogen, phosphorus and iron and sulfur, very important. So for example, we have nitrates, they will break nitrates, and if we have for example ammonia, they will break ammonia. Okay, so ultimately that ammonia will go into the soil, from soil it will go into the plants will die again, right? So that cycle goes from on and on, soil to plants and plants to soil, it goes on and on because of the bacteria. Heterotrophic bacteria, obviously they will be depending upon what? Other organisms. The majority of them, they are decomposers and they will depend upon dead and decaying organisms. Okay, Many of them, they have significant impact on humans. These are the benefits of the bacteria. And the first is what? Lactobacillus, it helps in formation of what? Curd. Lactobacillus, we can take example. Then we have production of antibiotics. Anna, we can take example of what? Streptococcus, it can produce the antibiotics. Fixing nitrogen in legumes, rhizobium. Rhizobium, you can take example. Okay, so basic things from NCERT, you can go through it. Yeah, some of the pathogens. Cholera, typhoid, tetanus, citrus canker. Cholera is caused by, I'll write it here for the idea, Vibrio cholera. Typhoid is caused by Salmonella typhi. Tetanus is caused by Clostridium. Tetany. Citrus canker is caused by Xanthomonas. Okay, citri. So Xanthomonas name you can remember. Bacteria, they reproduce mainly by what? Fissions, binary fission or multiple fissions. But majorly they, unfavorable condition they will produce what? Spores. Okay. Under unfavorable condition they will produce spores. Spores are nothing but hard wall structure or we can say structure surrounded by thick wall. Then they can also reproduce by sexual reproduction by adopting what? Primitive type of DNA transfer from one bacterium to another bacterium. You can see here, actually it is not completely like this. For example, we have one bacteria here. Okay, they will make a conjugation tube. Okay, with help of villi. This tube is called what? Conjugation tube. Okay, we have DNA here. It will replicate, it will move from the tube and it will come to this side. Usually in case of sexual reproduction, it is not the main DNA. Okay, it is the plasmid. It is the plasmid that is transferred from one DNA to another DNA. But conjugation is not important here. Just remember simple thing that there is formation of conjugation tube with help of villi. Okay, but in conjugation, our not main DNA but plasmid passes from one cell to another cell. Okay, so last group guys, in case of bacteria, is our mycoplasma. In mycoplasma, one thing that they don't have cell wall, right? So if they don't have cell wall, they will not have the specific structure. So they completely lack cell wall. They're the smallest living cells known and they can survive without oxygen. So they are anaerobic organisms. If we talk about archaebacteria is also, most of them, they are anaerobic because in harsh condition, in extreme conditions, right, oxygen is not completely available. For example, if we talk about methanogens, okay, methanogens, they are living inside gut of cattle. Oxygen availability is very less or minimum. So most of the archaebacteria, they are also anaerobic. You can mention that point in your paragraph. Many of mycoplasma are pathogenic in case of animals and plants. So this year also they did not ask about any disease. Okay, there is certain disease, potato spindle disease and all. Okay, that is caused by mycoplasma, and witch's broom is also a disease that is caused by mycoplasma. So it is not given an NCERT from last 3-4 years, or I think more than that. They have not asked any about disease of mycoplasma. So there is no need of reading it from any book. Just focus on the things which are given in NCERT. Okay. So today we are finishing up to mycoplasma. Tomorrow I'll start with protista and fungi will finish it. Plant animalia, you know, it is very easy. So tomorrow we'll finish protista, fungi and viruses. Okay. Like small thing, viruses, prions and viroids that we will cover. About plant and animal, we will cover in their respective chapters. Okay. So see you tomorrow guys. Take care. Bye-bye.